Radhe Radhe everyone, welcome to my YouTube channel Biology with Tricks. I hope you all guys doing well. In previous video I have discussed about the apoptosis in C. elegans. Today's I am taking up with aging of C. elegans, right? Before that I just want to tell you that please guys be focused and you can take a screenshot of this videos or the notes. These notes are very easy. No one is providing this type of data or information for CSIRnet, and I'll ensure you that this much data is enough for you guys to read out this topic only. This information is enough, more than enough. Okay, so please be focused. Okay, I'm starting with the aging of C. elegans. I'm showing here a diagram, but this diagram is not that much difficult, but you guys will understand this diagram after the discussion of the whole topic. So I'll be taking up with genes in aging. We have to focus on 4 points. Okay, 4 points like DNA repair enzyme, which are containing ROS generation, which is a reactive oxygen species, and second telomerase and P53, which is a part of DNA repair enzyme. Then we have to focus on protein of insulin signaling pathway. Third, we have protein in mtorch signaling pathway. Fourth is chromatin remodeling enzyme. Right? We're taking up with the DNA repair enzymes. It prevents senescence. Senescence is related to telomere shortening. If the telomerase enzyme is mutated or absent, this will result into the shortening of the telomere. What is telomere? Telomeres are the terminal end of our chromosomes. Our chromosomes containing a DNA content, I hope you guys know about it. If the telomerase is mutated, then telomere shortening will be occur. And DNA repair itself a repair enzyme which is repairing the function of the telomerase prevent the senescence. Right? If mutation in some genes they result into the progeria disease means older than age, like if a person have age of 18 years and he or she may be look like a 30 years, this is due to a mutation in a gene which is a laminin cause a progeria disease. I hope you guys watch a movie which is a pa in which the Amitabh Bachchan playing a role of patient's father. A patient has been suffering with a progeria disease. These diseases occur due to the mutation in the laminin gene. Okay. Then we have another phenomena, wear and tear. According to accumulation of small traumas, right? If there is accumulation of traumas, they will result into the point mutations and the functioning of enzyme decrease. If the functioning of enzyme decrease, then there is a faulty transcription or translation. Aging will be occur. Okay. This is about the DNA repair enzyme. We'll start up with the part A, which is a reactive oxygen species generation. The source of this is radiations and reactive oxygen species, basically produced by the normal metabolism, can oxidize damaged cell membrane and nucleic acids. Okay, they are produced by the mitochondria. If there is a mutation in mitochondria, there will be an increased amount of the reactive oxygen species, and this will release hydrogen peroxide radicals, which are very harmful to cell or a body of a person. For this, we use two antioxidants, catalase superoxide dimutase, which will degrade the reactive oxygen species and slow down the process of aging. We took a two things, control and a test. In control, we have used that normal level of catalase and superoxide dimutase antioxidants. What we have found that there is a aging which is occurring fast, and in test we took a overexpression of catalase and superoxide dimutase, which result into the destruction of a reactive oxygen species which is causing the aging, and now they are destructed or they are destroyed, they are damaged. This will result into the slow aging process. In overview, we can say that high reactive oxygen species result into the fast aging, while low reactive oxygen species result into the slow aging. Then at high antioxidant uses, slow aging and free radical number is decreased. If low antioxidants used, then fast aging and free radical number increased. Then we have a part B of DNA repair enzymes, which is telomerase and P53. Basically, telomerase, I have also given you the information regarding this, that telomerase preventing the shortening of telomere, which are the terminal end of the chromosome. If telomerase is mutated, this will result into the shortening of telomere, and which in returns result into the aging of a person. Okay. Similar to that, P53. P53 is a tumor suppressor gene, which is TSG, and regulator of cell divisions. If P53 is bind to a repressor, it remain in an inactive state, but once a damage to a DNA occurs, it gets separated from its repressor and causes apoptosis. Right? Means if telomere shorten, it is a part of a DNA, means chromosome containing a DNA content, and the end of terminal end of the chromosome gets shortened. This will result into the activation of P53. Right, when the P53 is activated, then our replication will be stopped and repair doesn't work. It result into the initiation of apoptosis. Right, if there is a decrease in number of telomere and stem cell, it will result into the cause of muscular dystrophy syndrome. Please do remember this statement also. Then we have case 1. If mice and human with the telomerase deficiency. If human and mice have a telomerase deficiency, definitely if telomerase is not working, then the telomere shortening occur and a person age prematurely. If he is of 18, he look like a 30-year person. Okay. Case 2. Overexpression of telomerase in senescent cell extend longevity in mice without increasing cancer. If the telomerase is overexpressed, it will increase the longevity but do not result into the increase in a cancer. Okay. Then we have a mTOR pathway. mTOR pathway promote translation of mRNA, right? It lower the longevity, means it is causing a aging. It is similar to that, right? Causing aging. Then we have, it is functioning like an insulin signaling pathway. If mTOR is activated, it will result into aging. If it is an inactivated, then it will decrease the process of aging, or aging cannot be occurred. Okay. Then we have a it most important statement. Please do focus on this statement. Dietary restrictions decrease mTOR activity. When there is a restriction on the diet, this will result into the decrease in mTOR activity. When mTOR activity decreases, when it's inactivated, so the process of aging will be slow. Mice having a low mTOR, longer life, autophagy, means mTOR is in a less number, it is inactivated state, there is a no aging that much occur, so longer lifespan. Control mice, mTOR number is high, aging will be fast. So yeah, mTOR is activated, it will result into the aging. If it is inactivated, will slow down the aging. Then we have a chromatin modifications. Serotonin aging and code histone acetylation silencing gene prevent aging. Means serotonin function is to prevent the aging. If serotonin is active, then there is a no aging. If serotonin is inactive, then there is a aging. Okay, this is about the whole topic. Now I'll show you the diagram that I have shown in a starting. Right, this diagram is here. This is showing in a 3 in C.Elegans, mouse, and Drosophila. The gene working in C.Elegan is DAF2, which is DAF2, and in mouse IGF-IR, and in Drosophila INR. If these three genes are activated, they result into the decrease in a lifespan and increase in aging. Right? They are inhibiting the DAF16, IGF-IR inhibiting FOXO, INR inhibiting DFOXO, right? And they result into aging and decrease in lifespan. If the DAF2, IGF-IR and INR inactivated, they will result into the activation of DAF16, FOXO and DFOXO, which will result into increase of the lifespan and increase the longevity of the organisms, right? This is the diagram. Please remember these key points. These are most important. If there is a loss of function of alleles in ISPs, formation of dural larva in environment, that I have earlier said, that if There is a inactivation of the DAF. This will result into entry into a dural phase. This is all about the aging of C. elegans. I hope you guys will like this lecture and thank you so much.